from University of Pittsburgh, and I study neuronal mechanism for natural decision making. But today, I am happy to expand my knowledge on the topic of our developing brain. And we have 3 speakers here. Each of them will give an interactive talk where speakers have 5 minutes to introduce their topic. And then we discuss for around 10 minutes. And for the audience, please feel free to ask any questions you have, even the ones that you think that are dumb, because my job today is to be the dumb guy for you. That's why that's the kind of exclusive service you get only during a pandemic, right? But on a serious note, the current pandemic has had a huge impact on neuroscience research, especially human behavioral research in neuroscience. And that's why a lot of scientists now resort to recruiting online subjects for their studies. But just how well can we do with online subjects compared to traditional lab studies? Our first speaker, Jack Morgan Mizell from University of Arizona, is going to tell us more. So Mizell, I'm going to stop my sharing so that you can get on stage. So the floor is yours.
Can everyone see my slides?
Yeah. Perfect.
Awesome. Okay, so today I'm going to talk about a pilot study I've ran this fall, taking this task that's been very important in the study of cognitive aging and also individual differences in that, and sort of decomposing some work in the medial temporal lobe circuitry in its regards to episodic memory, called the Mnemonic Similarity Task, or the MST. So to give you an example, I'm just going to have us do a little toy version real quick. Okay, so the task consists of 2 phases, an encoding and a test phase. In the encoding phase, people have to answer based on a series of objects they see, whether that object would typically occur inside or outside. So for example, probably outside. This one's a little tricky. And that one I would probably say inside. And the answers for this really don't matter too much. What matters is that you really consider these objects and commit them to memory. We then have a test phase in the task where you're asked to say whether an object is old, as in it occurred during that encoding phase exactly, similar, as in it's similar to something you saw during that encoding phase, or completely new, so something that has no relation to things you previously saw in that encoding phase. So let's do that real quick. So this is new. It has no relationship to those previous objects from the encoding. This one is an exact copy, so you would say old for this one. And this ability to sort of recognize those objects doesn't really go down with age. And then lastly, we have this, which is an ice cream cone that is similar, but not the same to what you've previously seen. And this ability to discriminate similar lures from objects you've seen in the past is pretty sensitive to cognitive aging. So taken from previous work and lab-based studies, usually consisting of about 20 to 30 people in each group, we can see on the left this lure discrimination, that similar discrimination goes down as people get older. And then we have on the right, the recognition index, that ability to recognize an object as old, stays relatively stationary as we get older. And I ran an online test using this exact paradigm. And was able to replicate these results, finding a selective effect in this lure discrimination on the left, where older adults are represented in blue and go down relative to young, while the recognition index remains relatively the same. And the selectivity of this effect is really nice because it can assuage some worries about engagement between age groups and things like that. But I went a step further, and I had a subset of these people who did this original task do a separate version of the task, where there's a 2-choice variant where you have to just designate whether the stimuli are old or new, lumping that similar stimuli with those new. And we were able to replicate the selectivity in the lure discrimination, but not in that recognition. So I have scores of individuals across both. So what I did is I actually mapped their behavior across both tasks. So each one of these boxes represents an individual person, and we found they do correlate, and they're specifically better correlating for the older adult group in blue relative to the young, perhaps speaking to something about task engagement over time, with maybe more young people dropping out due to chaotic environments or different things where they're doing, which I'm sure we're all familiar with, myself with a very antsy corgi who always wants to play fetch while I'm trying to do things. So conclusions from this. I have shown I can replicate a specific age effect within a task, this MST, across 2 variants, and that I can capture some meaningful individual differences in performance across these 2 variants. Now, like I said, this is sort of a primer building upon for my dissertation. So I want to run this in larger samples alongside other cognitive tests that should theoretically relate to this. And I also want to examine sequential effects in the task alongside other subtle effects that may be taking a role that previously are very to do lab environmentally for a number of effects. Thanks.
Thank you very much. I'm clapping here for the audience for you. So I'm sure the audience are taking time to type their questions and vote. And meanwhile, I'm just gonna ask a really obvious question. So there are certain limitations in methodology with online subjects. So, for example, you cannot put them in a scanner, you cannot track their eyes, reliably at least. But you clearly have a plan to offer us a deeper insight into the neurological mechanism of aging, right? So can you tell us more about the next steps?
Yeah, so I can give an example of something that I wanted to do with this. So my lab that I actually come from is very into computational modeling and decision making. And I'm very interested in sort of looking at episodic memory and its relationship to that, differences in decision making processes as people get older. And this MST has shown a relationship to both structural and functional to the lateral entorhinal cortex. With people, that relating there. But in addition, there's more and more work coming out with intertemporal choice, where you have to decide between something sooner, an option coming sooner, or a bigger reward coming later, that also is showing some relationship with lateral entorhinal cortex function. So while I totally agree that you can't put someone in a scanner in their own house or anything like that, we can sort of draw on that work to answer questions across larger samples in a much quicker way. So I would like to correlate behavior in that intertemporal choice versus this MST. It's just one example of where I see this going.
Yeah, I'm excited. I think there are like a dozen ways that I can think of right now that you can manipulate the image, for example. You can, manipulate the sequence of the image shown, and you can, do all those kinds of manipulations and build behavioral model, but build models to account for their behavior and things like that, right? Cool.
Yep, that's the goal.
Okay, brilliant. Any other questions from the audience or the panelists? The panelists, since you are already on stage, you can just unmute yourself and speak if you have any comments or questions. Okay, so if there are no other questions, we can move on to the next speaker. Awesome. Cool. We have just spent 10 minutes. It's going to be, I'm going to say it's probably going to be my very first session in my life that I moderated that I managed to keep on time. So you guys are doing great. Thanks. So yeah, we have another interactive talk on the aging brain. So Alessandro Maria Salvatella
Correct.
From Purdue, sorry I butchered your name, is going to talk about how machine learning and differential geometry can tell how old your brain is. And yeah, Alessandro, the stage is yours.
So thanks a lot for the nice introduction, and thanks a lot for the opportunity. So first I want to apologize because I'm a mathematician and not a neuroscientist. So forgive me if I say something which is nonsense from the neuroscience point of view. Please correct me. So this is actually a work that was done mainly by some students of mine during a class project. So Justin, Coatan, Peter, Maxwell, and Jucoin, and they published it in a Purdue undergraduate research journal. So the question is to try to understand if the brain can tell us how old we are. Okay. So, and we're trying to use a combination of machine learning and geometric tools. So, of course, there is the importance of the question given by the fact that the population is aging. There is an increase of neurodegenerative disease, burden into healthcare, and so on. So, so there is the Human Connectome project, which is a publicly funded study aiming at mapping the neural pathways of the brain, which is a very nice data set that my student worked on, and included 1,207 subjects with brain measurement and corresponding age range. Among those, there were, we analyzed only 1,113. And so the variables included were 56 volumes, 68 surface areas, 68 thicknesses, and they were divided only into 4 age groups, which were somehow young adults. Okay. So what is differential geometry? Well, this is a study of smooth objects which are invariant by smooth transformation. One of the most famous problems in differential geometry was the Poincare conjecture, which was solved by Perelman in 2003. And this is a guy which won a Fields Medal, won $1 million, and rejected declined both of the prizes. So another very famous problem is the isoperimetric problem, which is just trying to understand the relationship between the area and the perimeter of shapes. Okay, and there are instances of this in different dimensions. One important thing is the isoperimetric inequality, which states that the sphere has the smallest surface area per given volume once you scale according to the dimension. And one example to understand that there is no upper limit is to just think about a rectangle with fixed area 1 and sides which enlarge and decrease each other. I don't know how to explain that. And so the thing is that one can use this type of ratio as a proxy of how spherical a surface is. Okay, so there are other measures of folding, right? So the LGI, for example. In the data set, there were not full information about this. And so we only had thickness and surface area. So we used some sort of surrogate of the isoperimetric ratio to try to understand if there is a difference between age groups. So we found these possibly not super exciting results, namely that we were able to classify around 40% in a 4-way classification problem using the data available. But the interesting thing is that using simply this sort of surrogate of the isoperimetric ratio, we were not losing prediction accuracy, but we were somehow having some more explanation on the feature engineered in the process. So some comments. So we test accuracy using validation set approach. We did a meaningful dimensionality reduction of 64.5% of the variables without losing classification accuracy. And I want to mention that dimensionality reduction is not completely trivial because rational functions like that don't exactly fall down in the general theory of universal approximation of feed-forward neural networks. Some conclusions. So we attacked the brain age problem using geometric and machine learning methods. We analyzed the data set of young adults and with brain biometrics. And we showed that direct and feature engineering can provide explainable dimensionality reduction without losing in test accuracy. And of course, the brain problem is still is a very fascinating problem. Here are some activities in my lab where students and researchers can interact and have fun. And thank you for your attention. I hope I didn't say too many boring things.
No, not at all. This is super exciting. These interdisciplinary approaches are always, very eye-opening for a person like me. So you kind of skimmed through it. So as far as I know, what you also had on the slides, that the human connectome project were mainly data from healthy young adults. Is that right? Like 21 to 35 or something?
Yeah.
How do you think your findings would extrapolate to, either end to old age or younger age? Are there studies in the field that can give us a hint about it?
So there are two somehow follow-up studies that some of us are doing, and one is again the time evolution of the brain biometrics, so to check what happens through time. And the other thing is comparing healthy controls and people affected by mental disorders. For example, what we have in mind is schizophrenia, but could be other things. And using also different type of modalities. For example, instead of structural fMRI, using fMRI or DTI. For example, I think that is known that there are fractional isotropies that have some sort of parabolic trajectory through age or something like that. But of course, this is very, very interesting. And there are some mathematical difficulties in dealing with the complete structure of the, for example, the diffusion tensor, which can be overcome by using, for example, other tools from differential geometry. What I have in mind is called, in technical terms, fiber bundle, whatever it means. It's just something that you have some sort of matrix at each point, like the diffusion tensor, and this matrix varies in a smooth way along, for example, the tracts, 1 meter tracts and that type of thing, right? So you have to use this type of tools to take full advantage of the structure of diffusion tensor, for example.
Yeah, it's kind of amazing to see. It's not easy for me, at least, to have a very intuitive feeling about how. I know you came up with all these differential geometric measures, and it's very hard for me to connect it to any functionality explanation or mechanistic explanation. Do you have any guesses, like simulations at all?
Well, it's a very broad question. What I think, one thing that we notice here is try to find multiple measures of girification. So there is the local girification index, which is, of course, is a local measure. There are more global measures, and there are which come again from these mathematical tools. So and there are things which are a little bit in the middle, and so it's hard to say which one is the correct tool. And probably the usual answer is it depends. And of course, I think that there are tools which are available to mathematicians which are somehow unknown but relatively usable by neuroscientists. And so I think it's important to somehow communicate, because in that way one can the mathematician can learn what are the questions that people in neuroscience want to answer, and neuroscientists maybe can have access to tools which they didn't know that maybe existed, but they actually are known by us, let's say.
Yeah, I totally agree. So I know that these connectome data are hard to get, and we just have so much on humans. But I know that there are a bunch of studies that studied the connectomes for marmosets, for example, other species. Have you thought about applying your methods there?
So moving forward, there are a lot of options. That's actually something very interesting. And as you mentioned, right, in this data set, we didn't even have the exact ages. So we had some sort of age groups. And of course, that somehow is a limitation because those thresholds are maybe they're meaningful, but they can cause a lot of errors somehow, right? Because in the classification accuracy. Of course, one can weight the classification accuracy based on if you miss the age by a lot or not, but still that's the complication. We haven't tried on anything on species different from humans, so that we haven't done yet. But of course, that is important because then it also, if done in a certain way, can be related to evolution and other important questions. So, of course, that's a great suggestion.
Yeah, I know. I don't even know what kind of questions can be asked and answered by across species, but right. But yeah, it's just fascinating to see these methods, mathematical methods can somehow relate to how our brain develops. It's fascinating to me.
Yeah, I agree.
All right, so I don't know what's happening here. We have quite a lot of audience, and you guys are quite quiet for this session. So, anyways, if ever you have any questions, again, please type your questions in the chat window. And even if the speaker has already spoken, he or she will still be staying here, and they will still be able to answer the questions. And also, I just want to remind everybody, if you have any thoughts, if you have any comments on the Neuromatch conference, you may tweet with the hashtag. I think it's hashtag NM3. Is that Galana? Yeah, Galana will post that in the chat, the exact hashtag. Okay. Then I think we can move on to the next speaker. So thanks to Alessandro again. And so we have discussed quite a bit about the aging brain, right? So now let's shift gears and talk about the younger brains. So Yvette Planel Mendez is a graduate student with Sabine Castaner in Princeton, a lab that's well known for never stopped producing beautiful science. And I myself had the honor to visit the Castaner lab in 2016. And since then, a series of important work have further established the role of frontal-parietal thetaism in attention. And I'm more than excited to hear about what's new. So Yvette, the floor is yours.
Thank you for that introduction, and good afternoon, everyone. Today, I'll be sharing a project that I'm currently working on looking at the rhythmic properties of attention across development. Attention is crucial for selectively processing information in the visual field, and it has been classically viewed as a movable spotlight that focuses in on an area of interest. Recent research has shown that this spotlight isn't constant, but rather flickers, allowing you to flexibly alternate between periods of high perceptual sensitivity and periods of lower perceptual sensitivity. The latter period allows for a higher probability of shifting your attention towards something outside of your spotlight that might have become relevant. In adults, this rhythmicity of attention is in the theta range, or 3 to 8 hertz. This rhythmic property of attention is very useful. For example, if you are in your room working on your Where's Waldo puzzles, this rhythmicity allows you to rapidly alternate between perceptually sensitive periods during which you are more likely to find Waldo, and periods where you are more aware of your surroundings and are thereby able to notice if anyone is sneaking up on you. This process has been studied in non-human primates and adults, but to date, nothing is known about whether or not it is present from an early age or if it has a protracted development over the course of childhood. In this study, we're looking at children ages 6 to 8 and 11 to 13 to understand the development of this rhythmic property of attention. We hypothesize that the frequency of this rhythmicity becomes more adult-like with age. To understand this question, we've implemented a dot cloud task from Ayelet Landau's group. In this task, participants foveally attend to a drifting blue dot cloud stimulus. After a random interval of time, a drifting red dot cloud appears. The appearance of the red dot cloud functions as a cue, and 250 to 750 milliseconds after this cue onset, either the blue or the red dot cloud slightly decreases in color saturation. The participant must accurately detect this color change, and we perform a thresholding procedure with each participant to make the task challenging enough that they can do this accurately only about 75% of the time. The key part of this task that allows us to probe the rhythmicity is the cue target interval. By varying this period, we present the color change during different phases of perceptual sensitivity, and this is reflected in the participant's performance. To make the task engaging for kids, we've embedded it into a fun game where an astronaut named May is decoding secret messages on different planets. We first brought 12 children into the lab ranging from ages 8 to 15 for a pilot study to test the feasibility of this task. From this pilot, we learned that 360 trials was sufficient and doable for the kids. This takes about 25 minutes with explanations and breaks. We also found that different children needed vastly different numbers of trials for adequate thresholding. So we optimized our quest procedure by having it end whenever a participant reached a 95% confidence interval, rather than after a set number of trials. Finally, we experimented with providing auditory cues for correct and incorrect responses and found that, as seen in this figure, the response cues dramatically increased the amount of false alarms per participant. A false alarm is when a participant responds to trials where no color change was present, and this indicates that the participant might be guessing. Without auditory cues, there are barely any false alarms, making the data much more reliable. Next, we tested a small group of 5 children where we implemented the lessons learned in our pilot and analyzed the hit rates over the variable cue target intervals for the conditions where the blue cloud and the red cloud each underwent a color change. From looking at the hit rate, we can see a rhythmic pattern starting to emerge where some of the cue target intervals produced higher hit rates and others produced lower hit rates. To find the frequency at which this rhythmic oscillation is occurring, we perform Fourier transforms of the hit rates to find peak frequencies and calculated significance thresholds, the dotted lines, using permutation testing. Even though this is a small initial group of 5 children, we can see that, especially in the blue cloud, a peak is starting to emerge in the theta range. As we move forward with this study, we plan to expand into a school setting and collect sufficient data for both of our age groups. If we find that the rhythmicity of these 2 age groups is significantly different from each other, we plan to collect additional data for the ages in between in order to get a more holistic picture of the developmental trajectory of this process. Future directions potentially include using this paradigm to understand if this rhythmicity is altered in neurological conditions such as ADHD that present with atypical attention function. Finally, I'd just like to thank all of our participants, the Kastner Lab, and our funders, and thank you for listening.
Thank you very much, Yvette. Very interesting. I have like 100 questions, but yeah, I'll just go ahead and ask them. So first one is, I probably missed it. Why would you think the rhythmicity would change during development? What is the underlying hypothesis?
Yeah, so there's many studies that have shown that attention is basically not fully developed in children, and attention itself is still developing throughout childhood. Attentional processes are a little bit slower in younger children, and then they mature towards adult sort of standard. So that's one reason we think that perhaps this is probably reflected in these attentional rhythms. Also, just structural studies also show that areas that control attention are also not fully developed in children. So these different factors sort of lead us to believe that if attention isn't fully matured, that it's also possible that these rhythms aren't fully matured in children. And in terms of a specific hypothesis, we think that it's very possible that these rhythms aren't fully, that this rhythmicity isn't fully set in children yet. So this rhythmicity sort of represents a communication, sort of a control between sensory and motor areas, this allowing you to switch between sensory and motor control. So we think that perhaps we won't be able to detect a sort of developed rhythm in children just because they don't have this sort of developed connectivity yet. Or alternatively, the rhythm in children might be might just be a completely different rhythm than one of adults. So in ADHD, for example, there's different types of ADHD presents in different ways. So some children with ADHD present as being hyper-focused, where they get into these periods of being locked in, and other kids with ADHD are very hyperactive, where they're unable to lock in. So we think that perhaps in children with this hyper-focus, where they stay locked in, perhaps what they have is a very, very slow rhythm where they stay in this locked in phase and very rarely get the opportunity to sort of switch to something else that might be relevant. And in contrast, the ADHD children that present as more hyperactive might have a very fast switching rhythm that allows them to sort of not sustain attention for a long period of time.
Yeah, thanks. I think it makes sense to hypothesize that children are still suboptimal, let's say, in the sense of how they deploy attention. The mechanism hasn't fully matured yet. But I'm just thinking this rhythmicity, how much is it intrinsic to our circuit, the neuronal circuit that we have to live with, and how much is it dependent on the environment? Like you can totally imagine that I can totally design a task where the distractors would appear at a frequency or phase that this kind of theta rhythm is not optimal, right? So yeah, what do you think about it? I don't know. So where does why is this rhythmic activity so important? Is it to best capture the environment, or is it something intrinsic in our gene? Oh, well, I should say something intrinsic in our like neuronal hardware, that it has to produce something like theta for attention to function.
Yeah, I think, yeah, I don't know if I'm answering your question fully, but in a way, I think maybe a little bit of both, in that I think it's this sort of oscillation as a mechanism that your brain uses to coordinate connectivity and prioritize different regions. And I think this is sort of an intrinsic mechanism that the brain uses in a lot of different ways. And attention is one way in which it's using sort of oscillations to coordinate behavior, sort of in this case, by prioritizing either sensory perception, so attention, or motor activity, like the ability to move away and capture something in your surrounding that you might not, giving you the ability to sort of like look away and capture something else. But I also, but I think it also develops sort of in a way to best sort of to sort of give you like an optimal sort of response in a way, if that makes sense.
Yeah, I kind of get it. Thanks.
Mhm.
So there's a question from one of our panelists. So Jack Morgan, you want to unmute and ask the question yourself?
Sure. Great talk, Yvette. I guess I'm curious about sort of response times in this task. Like, would you expect that to relate to the theta rhythm in any way, or the emergence of the theta rhythm across development?
That's a really good question. So we do look at response times. We don't find it directly related to the theta rhythm, but we do use response times to sort of get rid of, participants that aren't performing well or participants that are guessing, but we don't really see this connection between if you're in a good phase, you respond quicker, and if you're in a bad phase, it takes longer. Yeah, we more see it if you're in a bad phase, they're just more likely to get it wrong and miss the response.
Thank you. Yeah, I'd just be curious with like a drift diffusion modeling or anything like that. If anything would emerge between sort of a good phase and bad phase in terms of response bias and things like that.
Yeah, that would be really interesting to look.
So there's another question from the audience. Jao asked, there is some evidence that infant-directed speech from mothers changes from a signal with more power on the delta band to a signal with more power in the theta band as infants get older. Do you think infants have a more delta-like attention span? So sort of, do you see that as an extrapolation to your data?
Oh, that's a really interesting question. And yeah, not something I'd really thought about, but I definitely think so. I definitely think that it's possible that in childhood, kids are using different frequencies. So I wouldn't think before they approach sort of like the theta band adult, sort of like fully mature frequency. So I wouldn't find it too surprising to find that if you could test this in infants, for example, that they would display a different frequency, possibly a delta frequency or something like that.
Yeah, I think it some way it relates back to my question, which is why is theta so special, right? So some previous literature has suggested because we know that eye movements are on a theta have theta rhythmicity, and when you look at covert spatial attention, for example, then very conceivably, because you have more or less the same circuit for oculomotor have for eye movements and covert spatial attention, then you are probably sampling back and forth in space with the eye movement circuit, and that's why you have the theta rhythmicity. But it's not the case at all in your task, right? It's they are at the same place. The two dot patches are in the same place, and you still see it.
Yeah. So that's something. Yeah, that's something that I think needs to be explored a little bit more. So we went. This is more commonly probed in sort of these covert attention tasks. Definitely not a foveal task. And the reason we ended up going with a foveal task for children is because it is a lot easier to perform the task. And we were happy to see that you can still capture this effect. But I think more work definitely needs to be done to sort of understand the difference between sort of what exactly is going on in this more feature-based attention versus like the spatial-based attention, and perhaps maybe what the differences in this rhythmicity might mean in these two different cases.
Yeah, speaking of that, the stimuli being foveal, how well is your eye tracking with the children? Like,
yeah, so we started, we're basically what I'm trying, our eye tracking isn't great. So we decided, we did a pilot study where we basically looked at the eye tracking and didn't find we were hoping that in the microsaccades, we would find
exactly.
Rhythmicity. But when we did it in this task, because it is foveal and because there's so much movement going in within the fovea, this like movement and grading dot clouds sort of provoke a lot of microsaccades. So we weren't able to capture any rhythmicity in the microsaccades because there's just so much stimuli-induced activity. So when we first found that in an adult pilot. And when we first brought these kids in, we decided against doing eye tracking for them for that reason, just because we didn't really find anything very interesting in the microsaccades. So we basically aren't tracking that anymore. And then a second reason being that because for this particular behavioral study, we want to collect just a large amount of data. We wanted to be able to go into schools, take these tasks into schools and collect large cohorts of children. For this behavioral study, it wasn't practical to also to expect that we'd also be able to do eye tracking in this phase of the experiment. We wouldn't be able to take sort of the eye tracking equipment into a school, for example. So for this phase, basically, we aren't eye tracking. Perhaps for a future study, if we decide to go in the direction of doing like EEG or something like that, then we might eye track with a smaller cohort of children that we bring into the lab.
Yeah, interesting. Thanks.
Mm-hmm.
Since that we still have a little bit of time, maybe we can do a very small scale panel discussion. What do you think? What do you each think is the most important questions facing developmental studies in the brain? And Jack Morgan, I sort of want to give you more time because you are kind of cut the most. So you want to start first?
I guess I could start with my bias towards behavior. Yeah, I think with a lot of these studies, the thing that I think will be really interesting is taking a lifespan approach. So tracking from early development to older adults. So you can do this like cohort wise. And this is kind of done a lot in like economic studies and things like that, where you take behavioral neuroeconomics, that kind of thing, where you track like individuals across the whole lifespan and look at how that changes the function of where they're at from adolescence to senescence, essentially. And I think that is a very good direction in terms of trying to piece apart behavior and then sort of tie it to sort of brain development.
Fully agreed. Anybody else have any ideas they want to share?
I guess one thing I'm really interested in is figuring out ways to bridge behavior and sort of directly connecting it to neural correlates of that behavior. And this is, I think this is particularly difficult in development just because the methods that we have are so limiting. So I think we have to get a little bit creative in how we do that.
Exactly. So Alessandro, as a non-neuroscientist, you want to offer your insight too?
It's very hard for me. No, but I can say from a math perspective, where are the differences when you include the time component? So so let's say there are stationary problems in math and evolutionary problems. Evolutionary problems have a time component. When you have a time component, there is an extra difficulty because you want to control, you want to have estimates of quantities across time. So, for example, techniques, statistical methods like those of linear regression, something like that, are way too poor to take in let's say enhance the effects, not enhance, to leverage the time component information because they're based on uncorrelated structures. Well, when you have things which arrive across time, you need to impose some sort of, you need to, of course, take in consideration correlation among observations, and then opens the field to completely different and more advanced methods. So from my perspective, those are more challenging from the math point of view. And then, of course, you guys have explained much in a lot of detail the importance from the neuroscience perspective. But I can say that from the mathematical point of view, there are extra difficulties and more interesting questions to try to answer.
All right, thank you. And I think we are perfectly on time to conclude this session. Thank you all for the three speakers. It was really a pleasure talking to all of you. And thank you for the audience for your attention. And the next bunch of sessions will start in 15 minutes, so you have some time for coffee. All right, have a nice day. Bye.
For today, and let's start. The first one will be Zoha Hamad from York University in Canada, and she will talk about the effects of cortical resection of visual pathways. And these effects actually will be kind of surprising from what one naively would expect. Zoha?
Thank you, Matteo. So I'll just get started. Okay. Hi, everyone. Like Matteo said, my name is Zoha. I'm a master's student at York University, and I'm going to be discussing some results that we have using a unilateral cortical resectioning patient. So unilateral cortical resectioning refers to a treatment for pharmacoresistant epilepsy. So when epilepsy and seizures caused by epilepsy are so great and there is no other treatment for it, individuals can go through surgery in which the part that's responsible for the epilepsy is lesioned out. You can see that these lesions can be varying per individual. However, these lesions provide us researchers an opportunity to understand the nature and extent of cortical organization. So using these patients in the past, specifically a 2019 paper, showed that following these lesions, specifically of the ventral visual pathway, you retain normal visual perceptual abilities. So using a series of high-level and low-level visual tests, this paper was able to show that perception is retained post-surgery when related to the ventral visual field. However, we know that the two visual systems hypothesis suggests that there's two pathways. We have the ventral system that's responsible for vision, and we have the dorsal pathway that's responsible for visual motor abilities. So the question that still remains is that do visual motor computations, which are carried out by the dorsal pathway, follow a similar behavioral pattern to the ventral visual pathway, in the sense that post surgery, our visual motor behavior is still retained similarly as visual perceptual abilities are. And to this end, in order to test this, we looked at the behavior of a 16-year-old female who suffered from epilepsy at the age of 7. She had a cortical resectioning at the age of 13, which resulted, her surgery resulted in a left posterior parietal and a occipital lobectomy. So her lesions were contained to the left the left hemisphere. However, they were both in the dorsal and the ventral pathway. So we were able to test TC using Ephron blocks. So Ephron blocks are a test that's been used a lot in the literature in order to characterize both visual motor and visual perceptual abilities. These are blocks that have the same surface area, texture, mass, and depth, but they vary in their width and their length. And they're used, like I said, in order to characterize both visual motor, so the dorsal pathway, and visual perceptual abilities, so the ventral pathway. All these blocks were presented in the central visual field. We used 14 control participants. And while they were doing both tasks, we would track their thumb and their index finger using an OptiTrack motion technology system. In order to characterize their visual motor abilities, we had a grasping trial in which they actually reached for the blocks. And in order to characterize their visual perceptual abilities, we had a manual estimation task in which they just tell us how large they believe the block to be. Following both of these tasks, we looked at the 3D trajectory of their index and their thumb. So we were looking at the maximum grip rapture. And what this refers to is when you're reaching for an object, you tend to make, you reach with your thumb and your index finger. And the largest aperture that you make between your thumb and your index finger is known to be sensitive to the real object size. And this is referred to as MGA, or maximum grip aperture. And this was the measure that we were looking at during the task. Following this, we looked at the correlation. Correlation refers to the average. We were correlating between the average MGA every participant had for each block size to the real object size. So you can see this participant would have something like an R value of 0.99. They had exactly a difference of 5 millimeters, they would have an R value of 1. So we were looking at this value to characterize their sensitivity to the object size. And what we see is that in their estimation left, we can see so over here we have the average for the controls. We have their standard error bars, and we have TC in the red. So TC was our patient that we were using. And you can see over here on the y-axis, we have their correlation. If you have a high sensitivity, it'll be represented by a higher value, and a lower value with a lower sensitivity will be reflected by a lower value. And you can see in her perceptual task for her left hand, she has something that's very comparable to the rest of the group. She clearly shows a sensitivity to the object size. Similarly with her right hand, we can see that again. With grasping left, though it's different, her sensitivity does fall a little bit, it's still comparable to the rest of the group. It's grasping with her right hand in which we can see a very clear deficit. She's much lower than her own performance and compared to the rest of the group. So we see this deficit over here. However, with JND, JND is a different measure that we used, which we're looking at the standard deviation each participant displays in their own measurements. So this is a within subject test that we were looking at. And we're looking at their own variability when reaching for the same object size. So with this, you can see a little different. If you have a high value, that actually reflects a lower sensitivity, and a lower value reflects a higher sensitivity. So you can see with her estimation left, she has something that's very comparable to us with the controls. Similarly with her perceptual task in her right hand, again, very comparable to the controls. In grasping left, though it does rise a little bit, it's still comparable to the rest of the group, and it's not significantly different. You can see that clear impairment once again in the JND using in her grasping right hand. So it's her visual motor abilities in her right hand that are clearly affected. And so what that shows is that her perceptual abilities, even though there is a ventral lesion in her ipsilesional hand, we can see that she performs very normally compared to the rest of the groups. We can see this using both of our measures. And her perceptual abilities, even in her contralational hands, is still retained. So her perceptual abilities in both hands are fine. It's only her contralational hand in her visual motor abilities where we see a clear impairment. And this could be evidence for a difference in the reorganization of the visual pathway. So between the dorsal and the ventral pathway, we see a difference in the way it reorganizes itself. This could be due to a greater degree of hemispherical specialization of the dorsal pathway, or because of a differential maturation rates of the two pathways, such that the dorsal pathway matures before the ventral pathway and has a shorter maturation rate. And yes, that is everything.
Thank you, Zoa. So it's question time, and I have a couple of questions. The first one is, do you by any chance have any diffusion data or complementary task on this subject? Because this is a very fascinating subject where I would be curious to see how the actual white matter pathways would look like.
Yeah, so I don't personally have that. We didn't test her myself. I know she has been tested in other studies as well. And I don't think she was tested. There was a white matter test that was done using epileptic patients post-surgery. I'm not sure if she was studied in that. I can get back to you on that, but I don't think she was. In the 2019 paper that I mentioned in the beginning, there was a series of tests that were done on her. They looked at facial recognition, object recognition. They had other perceptual tests as well. So she has been tested extensively in the fMRI. So there probably is data on her. I just don't have it myself.
Okay, oh, that's interesting. So one question from the participants, there is, I hope that I pronounced this right, Jerome Smith, that asks, is the deficit specific to grasp or any visual motor task?
Yeah, so her deficit is specifically just in her visual motor abilities, and that too in her contralateral hand. So her right hand, her lesions are in her left side of the brain, and then we see that reflected in her deficit in the right hand.
Okay. And I wanted to ask you something else. So I worked a bit in epilepsy, and usually the most patients would have resections like in the temporal lobe or in the frontal lobes. Mm-hmm. Is the parietal lobe and the visual areas a common area to resect, or is an actual very rare clinical case?
Yeah, so exactly what you're saying. It is very rare. So that's why there's not a lot of data reflected about the dorsal pathway. That's why this work is one of the first to look at the effects of the dorsal pathway. Like you said, the temporal lobe is what most commonly the cause of epilepsy. I think I could be wrong, but I think in 41% of all epileptic cases, the temporal lobe is what's responsible. And that's often where the lesion is. So that's why this paper extensively looked at just the ventral visual pathway, because that's still more common than lesions in the dorsal pathway. Patients in the dorsal pathway are rare, so that's why there's not a lot of evidence, not a lot of results pertaining to that area.
And is this any plan to follow up with this subject? Because the paper is from 2019, so I assume that now she will be 18 years old, so the brain is changing.
Yeah, I think she was, I could be wrong. I think she was 15 years at the age of 2019, and now she's 16. So she does live in the States. So whenever we get a chance to continue to test her again, that would be an interesting thing to look at. But as of yet, there's no plans to follow up looking at it. Yeah.
Okay. So participants, any other questions? Also the other panelists, if they are curious about something from this study.
I can ask an additional question.
Yeah. Please go
on. My name is Jeroen Smeets, if you want to know it, how to pronounce it.
My apologies, you're on.
Yes, but it was quite good. Thank you. I really liked your talk. And my question was about the framing of your talk. It was in terms of size and grip aperture. And I was wondering whether it is something about knowing the location of your digits or being able to move to a certain location. So you could test tasks like moving one finger to the other finger.
Yeah.
I think like that. That might be interesting to see where the visual motor deficit is.
That's a great point. Yeah, we are looking at kind of like a comparative, let me just pull it up if I have it. We are looking at a different measure, which looks at kind of how the group compares, how the participant compares to themselves when reaching for certain blocks. It's kind of like in fMRI, how you would use RSA to see how similar they are when we're comparing block 1 to block 2 to block 3 or block 4. It's still in the works, but yeah, like you were saying, there are different measures that we could use. So far, we've only used MGA, but we're hoping to expand.
Yeah. And one of the reasons for this is that I think that the idea that almost everybody has that grasping is based on the size of an object is in a way incorrect. It's based on moving your digits to locations which happen to be corresponding to the size of the object. But I can send you some references on this topic. But
yeah, that would be great.
Yeah, this patient is very interesting to tackle this problem further. So
yeah.
I'll send you an email for further discussion.
Looking forward to it. Sorry, Matteo, I think you're muted.
Thanks.
No problem.
Thanks, Zoa. I was saying if there is no further question, we can move on to the next speaker. So we are with the right timing. Thanks again, Zoa. It was very interesting.
Thank you.
Now our next speaker is Laura Quaia from, this will sound very bad for Hungarian people, Eötvös Loránd University. And this topic is very fascinating. It's basically bilingualism, but in dogs. And the most fascinating thing is that basically the dogs are awake and unrestrained, and that are all family dogs. So Laura, tell us about it.
Thank you. I have a question for you. Have you ever wondered if dogs can distinguish between Spanish and Hungarian? Maybe not, because it's a super specific question. But I did. Why? Because of Kun Kun. Kun Kun is a border collie, and he was born and lived in Mexico City for 5 years, and then he moved to Budapest. What do you think? Do you think that Kun Kun can notice that the Hungarian sounds different than Spanish or not? It could be yes, because, for example, babies can do it. And in a certain way, family dogs and babies are similar because both are surrounded by humans, and humans were talking and talking all the time. And in this example, dogs, they are in the same environment. But what happened with the dogs? All this speech is relevant or is only more as noise for them? So in this work, we test dogs' neural sensitivity to auditory regularities that characterize language. In this work, we use fragments from The Little Prince because, of course, the dogs can understand specific words as their names, the names of toys, commands. But here we can check if dogs can process in a certain way the language in general, how sounds the language. And we use 4 different kinds of stimuli. First, we have in this example in Spanish a familiar language. For example, here.
Sois como era mi zorro. No era más que un zorro semejante a 100,000 otros. Pero yo le hice mi amigo y ahora es único en el mundo.
And also in Hungarian.
Egy másik bolygón? Igen. Nekem egy búzatábláról nem jut eszembe semmi. Te vagy a hibás, mondtad.
And as control, we use the same fragments, but in a scrambled way. So this control stimuli maintain all the low-level properties, but it doesn't sound as language anymore. You can hear. And this is for the unfamiliar. So with this stimuli, we had 2 questions. The first one was about speech detection. So we compared the cerebral activity from both languages versus the scrambled stimuli. And then for looking for language discrimination, we are looking for cerebral regions that can discriminate between both languages, but not between both scrambled stimuli. And here are the results. In this experiment, participate 18 dogs. And first, I would like to focus in the red scale results. This is the primary auditory cortex and is where is the distinction between speech versus scrambled speech. So in these regions is the speech detection, but as you can see in this graph, in the primary auditory cortex is not a language discrimination. Here I showing the volt signal change for all the stimuli. As you can see, it is a difference in the response between speech versus scrambled, but not between the familiar and unfamiliar language. They have a similar response. But now, if we focus in the blue scale regions, this is the secondary auditory cortex that is more rostral and ventral than the primary auditory cortex in the dog brain, is the language discrimination. These regions can discriminate the cerebral patterns between familiar language versus unfamiliar language, but, and this is important, not between the scrambled stimuli. So indeed, no cerebral region can discriminate between the scrambled stimuli. As conclusion, we describe a distinct stage of language perception in dogs. First, the speech detection happened in the primary auditory cortex. And we suggest that this process is more about the naturalness processing. As you maybe remember, the scrambled stimuli sounds very weird. So maybe the dog brain is detecting this is not natural. It's not necessary that this is language, no language. It's more about this. And then the language discrimination in the secondary auditory cortex, we think is related with learning about auditory regularities. And indeed, it is pretty cool because we never teach to our dogs, okay, this is your familiar language. You need to learn something. And it's only by exposition to the environment that dogs learn this. So as an advice, remember, when you talk, dogs are listening. Thank you so much for this. And now I'm ready for the interactive part.
Thanks, Laura. I have to ask this. So since the dogs were free, how the experiment did work? So how you managed to have them in the scanner staying calm and managed to scan all these dogs?
This is super nice because all they are family dogs. So we usually contact to the families in parks or by recommendation. And there is many training sessions, but indeed dogs are super social and cooperative. So when they understand that you are asking to stay super still, they tend to do it. And also all the time inside the scanner is the owner and usually also the trainer. And in this way, the dog is, okay, if you say that it's safe and all seems true, we can do it.
Okay, so apparently they are better subjects than human subjects.
Yes.
Okay, so we have already 3 questions, so I will start with those. So the first one is from Shreya Mishra, and is, would you expect biological continuity of this phenomenon to other house pets such as birds, cats, and mice?
I think indeed this is a super interesting question because one of our hypotheses, and in general in this dog cognition, is that the dogs are special. Are special in an evolutive way because we share a lot of environment, and indeed the dogs, in comparison to other domestic animals, they were selected for cooperate with humans. So maybe for all, it's an hypothesis, and we are trying, not we, people are trying to test with, for example, with wolf or other species, but it seems that dogs, as they are for cooperate with humans, so we are important. So they are looking for cues to interpret us. So I will say that dogs are special, but in the first level about the speech detection, maybe it's a more shared trait, but it's only an hypothesis.
Yeah, it should be interesting because I think that last year a paper, probably it was Journal of Neuroscience, came out, and it was about cats and humans. And basically, they found out that cats are more attached to their humans than we thought. So I would be curious to see what happens with cats. So the next question is from Carolina Shimabukuro, and is, how long did it take to train all those dogs? Impressive work.
Thank you. And mainly it depends of the availability of the families. So if the family say, okay, 1 per week, I can do it, usually less than 3 months. And it's super easy. And I think with the dogs, in comparison with other species, is they need short sessions. So this is a little more session, but short, and then it's super nice and easy to continue work with them.
To follow up on this, I didn't ask, but were they normal fMRI sequences, or maybe were like the silent ones so the dogs wouldn't hear all those noises?
Usually we use with visual stimuli with the normal one, and indeed is the normal scanner. But in this specific experiment, we use sparse sampling. So it's less noise from the scanner and more silence to hear the stimuli. And it's super nice because, as you can see in this photo, the coil that we use is for babies. In reality, it's for the torso of babies, but it's super nice for the dogs because they put in this way.
It is also kind of peculiar that I asked a question that Clement Garin just asked, and you
Yes.
actually added about the coil, so basically we asked.
Also,
the
just to so, there is also a flex coil. Then they are two cycles, and it's super easy because you put in both, and it's super nice because it's easy for the dogs. Don't feel as trapped.
Yeah. And then the next question is from Alexei Krotov that says, great talk. I heard the idea before that dogs react mostly to sequences of vowels. Do you think that your findings support that claim regardless of the language?
I think not necessary our work because it's speech in general. And but I think it's interesting, but unfortunately with our data, we cannot say more with our specific. But of course, there are other studies that show that dogs can detect that one word is different from other only for one vowel. So I think it's super nice.
Okay. And then we have Minye Zan. Do the dogs move their ears in the scanner?
I think it's a little difficult because they are using earphones for protection about the noise as humans and also to present the stimuli. And it could be indeed, but we are using a threshold that maximum the dogs can move 3 millimeters. So I think it could be, but we are worried about the noise, so we are protecting, so we cannot speak.
Yeah, it makes sense. And then we have another question from Clement Garin, and is there a way to avoid the important artifacts in the frontal area in dogs' bold images? Did you have some kind of artifacts to get rid of?
We know. We didn't done, and this is indeed a super good question, and it's a problem in the dog field because for the, as you know, the air is introducing these artifacts. And we have, as humans, here air and a little in the frontal, but now imagining the dogs with this large nose, so this is a problem. But indeed, in Emory, in the Emory University, the Burns team, they are develop a specific sequence to try to avoid these artifacts. So we are here more interested in the auditory cortex, but we not, but other teams, yes, they are improving this.
Okay. Yeah, that's interesting. So now I have two questions from Dion Lu. I will try to make one so we don't go over time. So the first one is, how many dog species are included in the experiment? Is there some differences among dog species in the ability of language detection and discrimination? And then the other one is, we see there is a shift of the location of both the red region and blue region in speech detection and discrimination. Do you have some intuition on what is shifted and what might be the regions in between are responsible?
So in the first one, it was mainly the cooperative breeds, we say. So most of them, they are in a medium size, and the most they are border collies, honestly, because they are super easy to train, super easy. And it's difficult to know if they are difference in the ability because they are dogs. All dogs want to cooperate with humans, but also they are reports and it could be some differences between breeds. So here we are only focused in the cooperative ones, but I think I will hypothesize, my hypothesis is not, they are no difference because it's a more basic process. So okay. Maybe for learning specific words or what they do with this learning, it could be, but in this point, I think not. And the last one is about the inter areas. We are super sure about the primary auditory cortex. So we say, this is the primary auditory cortex, but in the other regions, it's a little more complex to determine what is secondary and not secondary. Mm-hmm. So we think that the main process is in this region, and is the real focus is in the primary auditory cortex for speech detection.
Okay, well, that's perfect.
And thank you so much.
Thanks, Laura. And let's move on to our third and last speaker, that is Valentina Borghesani, that moved from San Francisco to Université de Montréal. And we are still on language, but this time in humans, and it's about what happens in neurodegenerative disorders. Vale?
Perfect. Let me share my screen. Is it working? Perfect. Thank you very much, Matteo, for the introduction and for the two beautiful talks that preceded me. And I'm here today to share with you some of the latest insights that we gathered on the neurobiology of language, thanks to our approach that combines neuroimaging, specifically magnetoencephalography, and a clinical model, that of primary progressive aphasia. And primary progressive aphasia, or PPA, is an umbrella term for 3 different clinical syndromes in which patients progressively lose speech and language abilities due to the neurodegeneration of different left hemisphere language networks. So in the case of the so-called logopenic variant, or LVPPA, phonology and or short-term verbal memory deficits are associated with left temporoparietal atrophy. In the semantic variant, or SDPPA, anterior temporal lobe atrophy is associated with semantic deficits. And finally, in the non-fluent variant, or NFV-PPA, grammar and or speech motor deficits are associated with inferior frontal gyrus atrophy. So today, I will focus specifically on the semantic variant PPA and illustrate what we learned about the spatiotemporal features of language networks, given 3 key aspects of these patients' profile, namely that they misread irregular words, such as reading chore instead of choir. They can tell you whether in a given picture there's an animal or an object, but they cannot name it. And if asked to give a verb in response to a noun, such as, say, bike, they would produce a very common so-called light verb, such as to do, instead of something more specific as to ride. So in a recent published work, we show that reading irregular words in these patients is associated with over-recruitment of parietal cortices. And this fits nicely with the double route model of reading, which theorized the existence of a ventral lexicosemantic route that allows you to read correctly irregular words that you have to have learned by heart, and a dorsal route that is sublexical, phonological, that allows you to just translate from grapheme to phoneme based on the rules of your language. So in these patients, given their atrophy, given their damage on the ventral route, on the anterior temporal lobe, apparently they have to over-recruit the dorsal route to be able to read. In a second similar study that is currently under revision, we show that semantic patients can correctly classify images as belonging to either a living or a non-living category, even if they cannot name them. So they cannot name the item, they cannot tell you that it's a zebra, but it can tell you that it's probably a living being. And in the MEG data, we see that this is associated with greater activation over bilateral occipital cortices, as well as superior temporal gyrus, and somewhat inconsistent engagement of frontal regions. So the idea here is that the over-recruitment of these posterior occipital cortices might be linked to an over-reliance on perceptual feature as opposed to more conceptual one to solve the task. So as we said, they have this atrophy in the anterior temporal lobe, so they cannot rely on those conceptual features, but they still maintain the perceptual one. And finally, and this is very much preliminary data, we observed that when asked to produce a verb in response, like a verb that is semantically related to a given noun that you give as a prompt, semantic dementia patients produce a higher number of unrelated verbs, in particular, the so-called light verbs, such as to do, to make, to get, very generic one. And this is in contrast with the other two variants of primary progressive aphasia, in which we see a completely different error pattern. The logopenic variant patients have the lowest accuracy and produce the highest percentage of not a verb response. So they would produce a noun instead of a verb, maybe related, but not a verb. While the non-fluent patients have the best performance, and when they fail, it's just because they miss a trial. They don't give the answer. So taken together, our findings have important implication for the clinical phenotyping of these different variants of primary progressive aphasia, as well as for the theoretical model of the neurobiology of language, in particular, the results that I've showed you concerning semantics. So we have seen that semantic variant patients with given their ATL atrophy, have profound semantic deficits, which translate into an over-recruitment of phonological processes in dorsal regions to read irregular words, and an over-reliance on perceptual feature in posterior regions to classify items. So overall, these findings indicate a pervasive reorganization of the brain network in response to the neurodegeneration. And they show us how defective semantic representation can be compensated by enhancing perceptual processing in occipital regions and spared computation in dorsal networks. And with this, I would like to thank my collaborators, but especially our patients and their families for their time in our research. And I'm very happy to take any question.
Thank you, Valentina. I will start with questions. I'm curious because this is what these patients manifested is a lot similar to what happens with people that have lesions or resections in areas that are connected from those pathways. So I wanted to ask you, do you expect that the neurodegeneration is happening just in the gray matter or also in the white matter? For example, I think about the uncinate, the IFOF, and these pathways.
Absolutely. So with the semantic patients, we know that the atrophy starts in the gray matter of the anterior temporal lobe, and it spread posteriorly. And the more it spreads, the more it affects also the white matter path that are nearby. With the other variants of primary progressive aphasia, the involvement of the white matter is even bigger at already early in the disease. So definitely we have to look not only at gray matter, but also at white matter in these patients. Absolutely. But it originates in the gray matter.
No, that's very interesting. So we have a first question from Joao Araujo. Wouldn't you expect errors in grapheme phoneme decoding in PPA, given that you should have to access the auditory targets anyway? Or is it possible to access phonological representations without auditory cortex activations?
It's a very, very good question. And it's one of the things that makes interesting the comparison between the variants. So the semantic variants, which is the one I've studied reading in, since they have this lesion in the anterior temporal lobe, but the superior temporal and the parietal areas are spared, for them, the phonological processing is intact. So they don't make errors, phonological errors, because they don't have a lesion in that area. But in the other variant, the logopenic variant, where the atrophy is actually more prevalent, superior temporal and inferior parietal, in this variant, you do see those phonological errors because in those patients, the translation from grapheme and phoneme is affected.
Okay, that's very interesting. And to follow up on my previous question, of course, I don't know if you have enough patients to do this, but if you had enough patients, would you be able to stratify them depending on how long they have been in the disorder, and then to see if you start to see a difference in the MEG activations in the frontal that will start moving more backwards?
Absolutely, that's a wonderful question. That it is an important factor. How long they have, like the time since the diagnosis, how long they have been into the disease. The problem, as you hinted at, is that this is a rare disease, so we don't have enough patients to do group comparison at, let's say, 3 different stages of the disease. We do use a measure of their overall status, like a neuropsychological measure of their overall status as a correction for some of our analysis to try to regress out the factor disease duration. And in other studies that are not imaging based, well, we look at the behavioral, we do have enough patients. But for the imaging studies, it's really difficult to find patients that are functional enough to undergo MEG. So we just don't have enough data to do that. But it is definitely a variable of importance.
And I imagine that there is a similar problem if you would like to acquire also fMRI and DTI and more complex things.
So we do not get task-based fMRI in these patients. For most of them, we do have resting state fMRI and we do have DTI, because we acquire those when we get the T1, the map of that we use to compute the atrophy. So that's it's a very interesting data set, and we will eventually merge the things.
Okay, that sounds very interesting. Okay, so we have another question from Luis Fernando Zuluaga. Can you comment on some of the differences between logopenic variants in PPA versus Alzheimer patients with logopenia?
Yeah. So this is $100 million question. So the logopenic variant of PPA, 90% of the time is due to Alzheimer's disease. So the underlying pathology in the logopenic variant patients is Alzheimer's pathology. That for some reason that we still don't really know, affects in these particular patients the language network in the superior temporal and superior parietal areas before the hippocampus. So these patients don't have the typical memory problems that you see in the prototypical Alzheimer dementia patients, but they have logopenic variant PPA. But the underlying pathology is Alzheimer's disease. And this is one of the reasons why it's so important to do an accurate diagnosis of these patients, because once you put them in the right, under the right clinical label, logopenic variant PPA, then you are pretty much certain on what is the underlying pathology, and it's Alzheimer's. And this has been proven with PET imaging as well as post-mortem findings at autopsy.
Do you have a similar confound for the semantic variant? Are there some cases due to some other different pathology?
So the semantic one are, again, I would say even more strongly, like 98% of the time, TDP43C pathology. So it's a different kind of neurodegeneration due to the misfolding of a different protein. But it's again a pretty homogeneous group, pathologically speaking. The non-fluent variant PPA, the one that is more frontal, that's where we see more mixed pathologies. And it's harder, given the clinical label, to match it to the underlying neurodegeneration. But for semantic and for logopenic, it's almost clear-cut.
Okay. Is there any final question from any of the participants, also the previous panelists for Valentina? Actually, I asked it and I have the question. Again, from Joao Araújo. Would like to hear your thoughts on how studying a more semantic variation of PPA, studying a more semantic variation of PPA can help us to know more about the structure of a semantic system in the human brain, and what are the most striking insights we got from studying this type of PPA?
Yeah, I do think that the most striking result in absolute term is the importance of the anterior temporal lobe as a semantic hub. A lot of functional neuroimaging data findings in healthy subjects show us that a large distributed portion of the cortex responds to semantic stimuli. But the fact that it activates doesn't mean that it's necessary to process semantic. So what these patients are telling us is that while a lot of the cortex might be involved, this area, the anterior temporal lobe, is the critical one without which you have semantic deficits. And it doesn't happen with lesion in other part of the brain. And then when you start looking in detail at the kind of deficits that these patients have, we can try to learn more and more about how this semantic hub is organized. For example, we think by looking more carefully at the different fine-grained deficits that these patients have, comparing the ones that have more medial versus more lateral, more left versus more right atrophy, we can understand how semantic is organized in the anterior temporal lobe, because the computations that are building these representations are probably graded and distributed within the ATL.
Yeah, I think it's a great pathology model for studying that.
Very interesting, extremely interesting.
Okay, and now we are at 46, so it's almost perfect timing. So thank you all the panelists for their presentation, and thank all the participants for joining. And please go have a break, and then there will be other sessions, and in a bit will be over for Europe, but the US will go on. So let's see you